PiNa-Tec bio-nanotechnology. Here we want to show you how to screen for the best protein expression condition using the PIA-PINK detection in a protein immunodot assay. PIA-PINK protein immunodot assay. Reagent to detect your protein making is visible to the naked eye. Here's the protein you want to detect. Here the pink nanogold labeled secondary antibody and the specific primary. PIA-PINK binds the Fc part of the primary, leaving the specific parts freely accessible to visibly detect the target. With PIA-PINK protein immunodot assay, you can easily screen for different expression conditions like expression strain, medium components, vector constructs, induction conditions, additives, or others. Here we see the setup for an exemplary 96-well test expression plate with 4 different strains in 3 different media, tested for 4 different vector constructs, induced with 2 distinct compounds, which gives 96 expression conditions tested at once. Analysis of these 96 conditions with PIA-PINK immunodot assay is straightforward. Transfer 3 microliters of each lysate onto a nitrocellulose membrane. Then transfer 3 microliters of positive control and duplicate in an extra row. Also spot 3 microliters of a negative control. Finally, add 4 spots from diluted target control for quantification. For example, 1, 10, 50, and 100 picomole per microliter. When the transfer is completed, the nitrocellulose carries 104 sample spots, 96 from screening and 8 controls. Place the dried membrane into a clean tray. Add 10 ml of PIA-PINK block solution to cover the membrane. To saturate remaining protein binding sites, incubate the membrane in PIA blocking solution under constant shaking for 5 minutes. Then discard the blocking solution. To immunostain the protein, add 10 ml of the PIA-PINK staining solution, hybridized with 0.7 microgram of primary antibody to cover the membrane. Incubate under constant shaking for at least 1 hour at room temperature. The staining is a constant process that can be observed without interfering with the reaction. When sufficient staining is achieved, take the membrane out of the solution and dry it on filter paper. In particle immunoassay, the staining intensity correlates with the amount of target on the spot. Compare the expression results in relation to the standard protein intensity and select the best expression conditions. Enjoy the future of immunoassays and optimize your protein production yield with PIA-PINK. No intermediate washing, single antibody incubation, fast and comfortably sensitive, visible and quantitative. The example you have seen was generated with PIA-PINK-COMFORT. Hybrids with only 0.07 microgram primary per milliliter staining solution give visible signal from 1 picomole target within 60 minutes. Even faster is PIA-PINK speed, which requires 0.2 microgram primary per milliliter to give the same staining result within just 15 minutes. Both velocities are available in 3 specificities, binding primaries from rabbit, mouse, or human origin respectively. No matter which PIA-PINK solution you choose, structurally they are the same. PIA-PINK, the effective staining reagent, is based on gold nanoparticles and antibodies, which selectively bind IgGs at the Fc part. Your primary antibody is oriented outwards, leaving the specific N-termini free for target protein binding. PiNa-Tec bio-nanotechnology, managed by Katja and Andreas Werner. Happy to get in touch with you via info@pina-tech.de.